Our species tonight is the dotted blue. Oh no, not another buckwheat blue. You probably remember that we have two groups of buckwheat blues. These are the little blues that feed on the buckwheat plants. One is called the dotted blue group, and the other is called the square spotted blue group. And we've talked quite a bit in the past about the square spotted blue group. The dotted blue group has two species in Washington. There's the old name is the dotted blue. That's what the group is named after. It turned out to be a group of species instead of just one. But there is actually a dotted blue. Anybody know what the letters SS stand for?
Subspecies?
Hmm?
Subspecies?
Mm-mm. It's Latin.
Sensu stricto.
And that means?
In the strict sense.
In the strict sense. So this is not just the Euphalotes anapes group, this is the dotted blue. And we have one population that we know of in the state. This is a population that Bob Pyle found. It's up by Rimrock Lake in western Yakima County. The reason it's so restricted is, as far as we know in Washington at this point, there's only one host plant that it feeds on, and that's the bare stem desert buckwheat, or Eriogonum nudum, and that's only, well, it's one of the only places where the plant grows. This is a photograph that's from the Butterflies of America website, because I don't have a lot of good pictures of the species. The dotted blue group, all of the species that are in the dotted blue group tend to have small round spots. And the ones in the square spotted blue group, the Batoides group, have square blocky spots, especially on the forewing. But they also have, if you take a male and dissect the genitalia, they have very different genitalia. So they're clearly different species groups. And this is what the eggs of, well, all of the Euphalotes look like. They're very small eggs, only 0.6 of a millimeter. And they tend to have this, it's not real clear in this picture, but they tend to have this swirly, look like they've been twisted at the top. And the entry hole where the sperm goes in, it tends to be squarish. And so these eggs are not really too similar to other eggs of blues, coppers, or hair streaks to which they're most closely related. Here's the little first instar larva. And this is only 1 millimeter, just a hair over 1 millimeter long. So if you lined 25 of these up end to end, they would add up to an inch. So they're very small larvae when they first hatch.
Dave, is it really that color, the yellow and the blue? It's so good.
Yeah, they are this color, and the head tends to be this kind of a purplish color, fading down to solid black at the actual head. And in the first instar like this, with most larvae, the hairs or setae look proportionally very long, and they don't really get shorter as the larva grows, but the larva grows into them, and so they look shorter in subsequent instars. And here's the next instar, second instar. And you recall that instars are the body forms that exist before and after molts, just like crabs. They molt periodically, and each time they molt, the characters of the larvae change. You can see this one's started to develop some a red streak down the back and so forth. There's the third instar, and it changes quite a bit. The third instar develops all these neat black spots, some red spots. And notice that the setae are very restricted, all just pretty much around the periphery of the larva, and that's very typical of all the buckwheat blues, the square spotted and dotted. And here's the next instar, fourth instar. They only have 4 instars, but this is an early fourth instar. And then just before they're ready to pupate, they develop some very bright colors, or they usually do anyway. Notice that the colors on this larvae, notice how closely they match the colors on the buckwheat that they're feeding on. They sequester these pigments, and then the pigments show up in the bodies. Sometimes you'll get some of these species, not this one, but other species that feed on leaves instead of on the flowers, which have the pigment, and they won't have these red colors. They'll be very pale. Here's what the pupa looks like, very typical of pretty much all the lichenids. And here again, this is actually one of our dotted blues. This is from Rimrock Lake, and you can see it's a rather worn one. we really are very lacking on photographs of dotted blues or anoptes here in the state. But again, you can see that it has the small spots, and both members of the anoptes group, the orange spots tend to be separated in kind of V or chevron shaped, and they're not welded together into a band. That would be a character of the other group, the square spotted blue group. This is from Butterflies American webpage, and this is an adult male. Here's the kind of habitat where the host plant grows, and this is on the south fork of the south fork of the Teton River, south of Rimrock Lake. And this bank right here is where the plant grows. These are the plants. You can hardly see them because they kind of blend in. And looking closer, you see they're very open plants, very openly branched, and the flowers are small. And these little leaves down at the bottom, those basal leaves are all the leaves that they have. But the butterflies don't feed on the leaves, they feed on the flowers. Here's what the leaves look like. With these senesce early in the year, you can see something has been feeding on the leaves, but we don't think it's the dotted blues.
Could be the aqua.
It could be the aquilines, which are also found in the same area, the aquiline blues. Now, these are the females, again, from Butterflies of America. These are Andy Warren's photographs. These are the females of both species, the anoptes, which is the dotted blue, and the other member of this closely related group, the Columbia blue. Both of them are in the dotted blue group. And here's a bunch of ways that the two species are different. I'm just going to go on and then come back. These are the males, same sort of a layout. So let's go back and try to figure out how we tell these two things apart, because they do look pretty similar. Again, this is taken from Andy Warren's descriptions in the Butterflies of Oregon book. And he starts off with anoptes, the dotted blue, are darker blue above, while the Columbia you recall, is our very common species that grows out in the shrub steppe, and anoptes is somewhat more montane. And anoptes, the dotted blue, the males are darker blue, and in Columbia, the males are more lustrous blue. And here we see what they're talking about. Here's the darker blue anoptes, and here's the more lustrous blue Columbia blue. So that's what he's looking at there. Next one is with anoptes, the dotted blue, the males have a broad, dark margin on the wings, on the upper surface. And in the other species, Columbia, the males have a narrow, dark margin. Here again, here's the dotted blue with the broad black border, and here's Columbia blue with a considerably more narrow black border. Ventral spots are small and poorly developed in anoptes, and they're somewhat more bold in Columbia. And this is true of both sexes. Here's the males, and again, you can see here in dotted blue, the anoptes, the spots are small and poorly developed, and in the Columbia blue, they're somewhat more bold. And if we back off and look at the females, we see the same thing. Here's anoptes with the pretty much small spots, and with Columbia blue, they're bolder, especially in the area around the orange aurorae. Okay, and that brings us to the next character, the orange hind wing aurorae. In anoptes, they went up in the up by Rimrock Lake, the aurorae, the hind wing orange band is pretty subdued, while in Columbia it's quite a bit bolder. And here again, here's anoptes. Look at the subdued orange band, and then Columbia blue is quite a bit bolder. That's true on the underside too. Anoptes has a pretty limited band, and then Columbia is quite a bit heavier. And then the next one we can't tell from these photographs, but the anoptes is somewhat smaller than the Columbia blue. Now the next one is one I have trouble with. John, maybe you can help us with this, but Andy points out that in anoptes, the wing outline is rounded, more rounded And in Columbia blue, the wing outline is more angular. Well, let's look at the males. So this one is supposed to be more angular, and this one's supposed to be more rounded. I don't see that too well.
Yeah, I don't either, none of those images.
Not in this particular set of them.
I'm glad you told me that Andy did this.
And here's the female of the same thing. And maybe it looks a little
It's
rounded,
a little more,
pudgy.
it's a little more rounded.
Yeah, but they're pretty similar. So I have trouble with that particular feature.
So often wing shape is a product of upbringing. I mean, if you have a luxuriant food plant and you feed really well, then you'll have maybe a more well-developed wing. I think those wing characters are sometimes pretty sketchy.
Okay, okay. The last one is the host plant differences. As far as we know, Nunnalle uses only one host in Washington. That's the bare stem buckwheat Ariagonum nudum. The Columbia blue uses two primary plants, compositum, which is the northern buckwheat, elatum, which is the tall buckwheat, and sometimes strictum, the stick buckwheat. In Oregon, they do report that Nunnalle also uses elatum, the tall buckwheat. So we need to kind of keep our eyes open in Washington to see if we might possibly find more populations of Nunnalle. But right now, this is all we have. And if you see these things in the field, please try to get some pictures, because as you can see, my pictures are not very sterling. These are all Andy Warren's pictures. And that's all I have. Any questions? Rod?
And it seems to me that, except for the food plant, every one of those differences is
Fuzzy.
just an average kind of a difference that could be in some specimens completely swamped by individual variation. So who proved that these are different species? Did they do DNA or what?
Well, actually, to answer the question, you're absolutely right. Almost anything that you measure, if it's subject to variability, any distinctiveness can be swamped by that. Bimodality then will get swamped by extreme variability. But in this particular case, yeah, molecular work has been done between these two, and they are segregable. I mean, there are haplotypes that aren't shared, but I think that, these are really tough, and most of the evidence that people have is actually in the field evidence and observing these things and seeing the close association. For instance, in the Anoptes, this association with Ariagonum nudum in California and Oregon is so extensive, so widespread, it crosses over all other species of Euphalotes, that itself is significant, the fact that it's maintained here. I think the actual phenotype, the problem with butterflies is that they're just flying billboards, okay? And we
Flying
see
what?
billboards.
Billboards, okay.
Yeah, they see, what we see is what they're telling us, okay?
Oh, okay.
Well, let me remind you of something. Monarch Viceroy, they lie to us all the time. Calico owl butterflies, they lie to us all the time. Any of the crypsis, any of the patterns that, enable butterflies to hide out from us are deception on the wing. So, people that look really hard at wing characters and think they see something, mostly are saying something that doesn't exist. I mean, they're rarely, I'm going to go into coleus and probably undermining my own presentation,
But with any species, you're always looking for a combination of characters. And as you say, any one of these characters can be blurred out or even lacking in an individual specimen. But if you look for this set of characters, you can usually separate these things.
I know that back in the late 1990s, I was collecting in Southwest Oregon, and whereas in Washington, I was able to usually distinguish between species of Euphalotes. I could not, for my life, distinguish down there. They were that much different down there from up here.
Yeah.
But I have a really ignorant question. That is how old is, or how long have people been using this name Columbiate for?
about 10 years.
How about that?
It was 2005, almost 10 years.
Was Andy the first one to raise it to the species level?
Yeah.
And Columbia exists as a subspecies part of it. This is Andy Warren when he did his butterflies for Oregon. it's a book with a lot of text and no pictures, but it's good.
All the
Jim?
best books.
In Washington or any other states, do they overlap in some areas, like where the shrub step comes into the ponderosa pine? And
Not that we found, we only really have one population of Monopoly so far, and it's the one around Rimrock Lake, and we have not found a Columbier there, but it's not too far away.
When we're on a field trip, what do we call these two now?
Dotted blue
We still call it dotted?
and Columbia blue. Yeah, dotted blue has always been a legitimate name, but we didn't think we had it in Washington. When they started splitting up dotted blue, we thought it was only further south, but then Bob Pyle went out, well, it's actually been 10 years ago now, but Bob Pyle went out and discovered it here in Washington. So we do have dotted blue. In the past, we thought we had lots of dotted blues all over the state. But now that we've learned more about this genus, we realize, oh, they're all this Columbia blue.
On the question of, are they really different, doesn't the genitalia difference? You
No,
say
not
they
in-
are definitely?
Not in these two. These are both in the same group.
Okay.
So the genitalia are pretty similar. They're very different from the square spotted blue group, easily separated. But these two, I don't think you can really separate.
Their food plots are different, very different, generally, mostly.
As far as we know, yeah. But again, we just need to keep our eyes open and see if we don't start finding monopolies in other places here in Washington. We might.
There are places in Oregon where both of these are sympatric. You know, a lot of feeders and nudum feeders. They're displaced in time, somewhat asynchronic, but they're in the same places.
Okay. I
Dave, can you tell us any of these field trips that people might go on that could see either of those?
Well, we'll see Columbia blue just about everywhere. And we are having one trip late in the season up to the top of Bethel Ridge. We could see anoptes up in that area. But during the year, we'll see Columbia blue in quite a few of these spots. And a whole lot of other good things.
I think we saw a dotted blue at Research Canyon last year.
Yeah. It would be on the, above the springs, Garrison Springs.
I have a photo of one laying eggs.
Yeah. It's certainly in that vicinity.
That's a real dotted blue?
You ID'd it for me.
Well, it was like one of those concessions you made to nomenclature on a field trip.
I might have said dotted blue group, I hope.
That's what it was.
Okay, thank you.